Tonight, I want to welcome Max Plikus here. Dr. Plikus is a long-time member of the Stem Cell Research Center, where was recruited here as an assistant professor, is now very important on campus, really critical in a Chan Zuckerberg initiative, a P30 on skin and wound healing, a training grant also that is based in that area. He's going to tell us today about stem cell plasticity in response to tissue injury. Sorry, we were talking engineering over here a second ago.
I'm okay with engineering.
And I'll highlight that we're doing something a little bit different the last couple of lectures, and that's pairing a basic science faculty member with either a student or a trainee of some kind. So our second lecturer this evening, linked to Max, is a clinical fellow that is working on her training, completing her training as an MD PhD, and now doing a research fellowship in Max's lab, Inti Leo. And she is going to tell us about her work that is linked also to tissue responses to injury. So thank you very much.
All right, here we go.
And special thanks to you to show up here. What is it today? Tuesday. So your Tuesday evening, you're dedicated to this. Thank you. Eileen, thank you for your introduction. Like I said, I've been here for 10 years now, so it's starting to feel like a long time. I only wonder, what am I looking at? So I'm going to remove the text. So what do you guys think you are looking at? Like, what is it? Engineers in the room, what do you guys think this is? Yeah.
Is it cheating if I know already?
I teach this class to like the computational students, and every time they look at the biological slides in the journal club, I see this blank look. So I just ask them, and then I realize they don't understand what they're looking at. Okay, so I see.
Map of Ukraine.
It's Crimea, actually, right there. So, okay, so just to clarify, right, so the lay of the land. This is what we call like a whole mount image. It's actually a piece of skin taken from the mouse that was wounded. So the thing in the middle is scar, okay? So it looks white, it's like very, a lot of collagen. The stuff on the outside that's brightly colored, the black spots are hairs that have melanin, so they're pigmented hairs. The orangey stuff, I think it's better seen like around the top and on the right. This is actually purposefully stained fat cells, so it's lipid. So normal skin actually is really complex. It's packed with hair follicles in the mouse. I mean, if you think of a mouse, a lot of fur. And underneath that fur, there's actually like a layer of fat cells. So if you were to wound skin in a mouse, as it heals, it's unable normally to restore any of that complexity, right? So it's unable to regenerate. Mice are mammals, we're mammals. We unable generally to heal by regenerating back what was lost, right? We replace the lost tissue with a substitute tissue that's called scar. And clinically, scars, of course, is a big problem. You know, there's probably a whole big book written on scars of all sorts. So now, people understand that wound healing is a problem. So in pop culture, in movies, if you're a superhero and you got some burn wounds, deep full thickness burn wounds, the way you heal them is by, of course, setting them ablaze, and boom, miraculously it heals, right? And notice arm hair is restored regardless of the fire. So of course, you know, that's the superheroes. In reality, something like that would not look very good. So this is an example of something that's really an intractable problem in wound healing, chronic wounds, especially in people with diabetes, type 2 diabetes. So a lot of times they appear on feet. They don't heal. This is a problem that we still have not solved. I apologize if you haven't had dinner, but some of these wounds could look very gruesome. So if they penetrate deeper, like here, so the tendons and bones are exposed, you can develop bacterial infection of the bone. It's life-threatening. The only currently accepted medical treatment for this is exactly what? It's amputation. So, and that means permanent disability, so huge burden on medical system, happening all the time. So if we can learn to treat that, that's a huge savings. Now, wouldn't it be nice to regenerate the foot back? It would be nice. So back to Hollywood.
So take a look here. So essentially, there's an lost arm, right? So this is like a scientist who is obsessed with research that will benefit him. So he wants to identify regeneration signals that typically exist, for example, in lizards that could grow tails back. You know, you live in California, you see lizards growing their tails all the time now because they warmed up, so now they're all out. Okay, so what they used is what's called transspecies genetics. So he took a gene from a lizard, gene A, and injected into the stump. Next thing happened, it worked. The arms start growing back. But like with any medical trial, there are side effects. So skin started turning lizard-like with scales. Side effects became systemic. And of course, he turned into a villain, right? So now, in reality, can animals regenerate arms? Yes, they can. So if you take a salamander, specifically axolotl, which is a commonly used research model, it's an amphibian. If you cut off an arm, as shown down at the bottom, this is actually just like a photoshopped images of consecutive regeneration of an amputated arm. So you start here, you cut it off, then it grows back. It's like magic, but it's not. It's true biology of these animals. Notice here, so there's another salamander. It just lacks pigment, so it's kind of whitish. There's 2 arms growing there. So without going into details, scientists could trick the shoulder, small tiny wound in the shoulder, to believe there is full amputation, and this would induce regeneration of the entire arm, right? So I think this really tells us that there are animals out there, they're typically not mammals, that have this profound regenerative abilities, right? Every time somebody discovers a new factor promoting this, it's like a Nature paper, science paper right away with the cover image, because it's just so fascinating for people that this is possible while we can't do it, right? So back to Earth, so we mammals, right? So what are we capable of? Okay, so it's sort of well known that in mammals during fetal time, so in utero, if you're like a late stage embryo, fetus, tissues heal, so to speak, scarlessly. So this is an example of a mouse experiment where wound was introduced to skin during embryonic day 15. It's about 2/3 into the development of the mouse. And you can't tell what the wound is, and that's the image on the left, because it healed scarlessly. Day before birth, approximately embryonic day 18, if you were to do the same kind of wound, and then the pup will be born, you can immediately tell the difference because it doesn't look the same, right? So now, just a few days later, you start to heal by scarring. So there's something that earlier fetus had that the later fetus has lost, and as an adult, you also don't regain that ability, typically. So the idea that has been formulated is that, well, we just need to learn what those early stage fetal skin can do. And we kind of go back in time, I call it go back in morphogenesis, and recapitulate the embryonic program for scarless healing, and then possibly even kind of reignite it in an adult like us, so we can do that. So very interestingly, at this point, probably a decade and a half ago, it has been discovered that if you were to take an adult mouse, and if you were to introduce wound that's slightly bigger than a typical wound that is done in experiments, but still survivable, these wounds will heal, but instead of just forming scar tissue, and now you're looking actually at the piece of the skin that's not inverted under the microscope. So what you're seeing is normal hairs at the edges, they're black. Then you have a little bit like scar tissue, but right in the middle, all this stuff is brand new hair. In the center of the wound, there's actually hundreds of them that have spontaneously regenerated. There was no biomedical engineering involved, no biomaterials were placed, no molecules were placed. This is an endogenous capacity of this mouse to respond to large wound by regenerating brand new hair. So who in the audience would want to have hair growing out of their wounds? Yes, yeah. On a scalp, it would be nice, right? Now, like somewhere on the forehead, like Harry Potter, maybe not so much, right? So the answer really depends where. Now, but why would animals in general want to have that ability? We need to think outside of ourselves, right? So if you're like an Arctic animal, like this Arctic fox, and you get into a fight with your neighbor and they like rip a big giant hole out of your skin and then you heal, but it's minus 50 Celsius outside, if that's a denuded piece of the skin, you will probably die from hypothermia, from something like a centimeter-sized scar. So regenerating new hair to provide thermoprotective layer of the hair would be actually pretty linked to survival. So there's deep biological need for many furry animals, mammals, to be able to do that kind of stuff. But what this also tells us is that adult cells in certain kinds of wounds, like the large wounds that I showed you in the mouse, are able to enter like a more plastic state without any exogenous molecules being applied. No fancy engineering, they just naturally do it. So there's an endogenous program that can be unlocked. So if we study it, we could understand some really deep biological capacity of mammalian adult cells to re-engage in regenerative healing. So shown here is actually, like pieces of the figure from my good colleague Mayumi Ito. She's at NYU right now. We trained previously together at Penn in the same lab. So she's the one who kind of rediscovered something that was first done in 1949. Like first time this ability to grow new hair was discovered in rabbits in 1949 and then forgotten for decades and then rediscovered in 2007 in mice, a relatively accidental finding. So if you were to make wounds that are relatively big, in the center, a brand new hair follicles will start to spontaneously form. Thinking of a hair follicle, think of like an onion growing in the ground. It's actually just a bulb that just extends deep, and then eventually like a hair will come out of it. So what you're looking at here is consecutive pictures at the top and numbers of days after wounding. So on day 15, the new little buds start to appear. Give it a couple of days, they will start to extend deep into the skin. This is hair follicle. And then by day 80, that entire wound is covered with a tuft of new white hair. It typically lacks pigment, but in fact, that's how we can tell it's different, but it's a hair. By all other means, it's a normal hair, just white, okay? And they are right here in the center. This is what's called histological section. So you can actually see these little squiggly things. This is parts of the hairs that have regenerated. Other than hairs, there's something else that regenerates that's pretty fundamental, actually, for thermoregulation, is fat. So our skin has this layer of adipose tissue, just kind of known as fat cells. What you're looking at here is staining, and now again, inverted skin upside down, looking upside down at skin, of the center of the scar that was stained with this lipid dye that's bright orange. And what you can appreciate here is that there's this like two islands in the center, right? There's this white streak, so each one of the streaks is that like unpigmented hair. But they're like swimming, so to speak, in this carpet of fat cells. So, and if I were to zoom at the top left corner, you could appreciate normal hair and normal fat, scar, how different it is, and the newly regenerated hair and fat in here, bottom right. This and this look equally good. So what mice are able to regenerate spontaneously is as good as what they formed as fetus stage. So that's very fascinating. So what we are interested in many aspects, like what are the molecules involved, like how do cells become this way? So I'll just tell you just a tiny bit. So before they become new fat cells, the progenitor cells behave like a typical scar-making cell. We call the cells myofibroblast. Myo from muscle and fibroblast is a fibroblast. So myofibroblast is muscle-like fibroblast. So essentially it's a contractile cell that turns on this like ability to contract. We can label them conveniently. So here what you're actually looking at the top of the wound that was stained, and it's a genetically, it's in the mouse that genetically modified to be able to detect this contractile wound fibroblast. They typically are culprits of scarring. They actually, previously at least, they were thought to be this like pathological cells. Like you want to get rid of them. You want to minimize the ability to contract. But what we and I mean, this is actually summarized here. So your normal benevolent skin fibroblasts after wounding get triggered, they get excited, they convert into this kind of almost pathological state called myofibroblast. They start to produce a lot of collagen, they pull on it, so everything gets kind of distorted around them, and thus scar forms. So that was sort of the conventional view, but what our research has shown is that they only do it for a while. They don't continue to do it forever and ever. They actually do it just for about 10 days during early phase of scar formation. Then they could switch off to start converting into new fat cells. So this new fat cells, so there's this little area here that's magnified here, and arrowheads point at the few fat cells. And if you were to like tease them out and put them on a cover slide and kind of look at them deeper, so you would see the lipid droplets and all of this blue genetic marking. So this is like fat cells that have derived from contractile wound fibroblasts. So that's kind of an evidence that previously they were these bed cells, contractile cells, but now they became fat cells, which is quite desirable from many points of view. So now get ready, this is a little bit deeper science. So we can profile the molecular signature. So we use this technique called RNA sequencing. It's actually quite easy to appreciate. So if this is like Y and X axes, so these circles, they actually represent like gene expression state of this wound fibroblasts at different days, and they label day 12, then 15, day 21, day 26, so consecutive days. You can clearly see that what we call replicates, they're really close to each other. But depending on the time point after wounding, they keep changing. So when they move around along the XY axis, that means that the gene expression dramatically changes. And we can then kind of very carefully look at various type of genes, and a lot of the stuff actually makes a lot of sense to us. So I'll just highlight to you the fact that so genes, typically they're like muscle-like contractile genes, they are turned on early. So like at day 12, they will be really contractile. But later days, by day 21, they all but shut it down. So there's no more contractile gene expression. So they stop being contractile. That's kind of proving my earlier point. And then a lot of other interesting things happen with other type of molecules that basically tell us more of the story, what happens to them next. So we dive deep into these genes and we kind of look for clues to how do they become fat cells. And the answer is the following. So they activate like deeply conserved molecular program for how embryonic fibroblasts become fat cells. There's actually, it's well worked out in an embryo how a fibroblast become fat cell in the body. And there's actually a series of consecutive molecular factors, we call them transcription factors, that get turned on. And transcription factor is very important because it's almost like a master key to a lot of gene expression. If it's on, then there's a lot of like genes under its control that get turned on. So the earliest factor that gets turned on in this very linear program. So we start with, say, an embryonic, let's just say this is embryonic fibroblast, and this is a mature fat cell, and all of these names that make not much sense to you for an adipocyte person would be like super important genes, right? So the first one in them is this molecule that has this like not a very pretty name. It's called ZFP423. Stands for zinc finger protein 423. It is the master regulator of fat cell fate. So it gets reactivated in wounds. So in normal, so when you see this blue cell, so it's a genetic marking of that molecular expression, when fat cells normally form in skin of a mouse, in an adult mouse, you actually don't see it. So no new fat cells form. But after wounding, this process gets reactivated, and all of a sudden you will start seeing all these new fat cells form. So I actually have this image, I really like it, so I'm gonna like zoom on it to kind of like demonstrate my point. So it almost looks like a little piece of art, you know, a little abstract. So this kind of shadowy, long, squiggly things, these are hair follicles. The little kind of dot pattern, if somebody splattered paint on it, each individual dot is a new myofibroblast that is now acquiring new identity of a fat progenitor. So we caught it in the earliest moment of them trying to become a new fat cell. That's what it is. And here is the outcome of it. So if we were to look at later wounds, so normally, for example, if we take a control mouse wound, we will have all of this dark orange circular fat cells. They are not there in mouse that is mutant for this molecule. So this picture is a little bit more complicated than what I just told you, because like you would say, like, wait a second, but I see these blobs of orange. Now, this is oil gland of a hair. It also is oil containing, so it will get stained, but they look very different. They're big and blobby, but individual fat cells are much smaller and intensity of orange is much stronger. So you just kind of have to take my word for it. So this might show inability to regenerate fat, even though they regenerate hair. So this might regenerate hair, no problem, but not fat. So normally what happens is that fat cells can become, so myofibroblasts become fat cells if they are given a trigger molecule. The trigger molecule comes from hairs that regenerated a few days before them. That trigger molecule is called BMP. So BMP is an abbreviation. It means bone morphogenetic protein. So hairs, when they grow in a scar, they produce a lot of this molecule. Surrounding myofibroblasts, when they see it, that triggers them to activate this adipogenic program that's dependent on the ZFP molecule. And it's an in vitro experiment. So here we cultured scar cells from a much earlier wound when they have never seen BMP. And if we attempt to make fat cells out of them in a dish, they just don't respond. They just continue to be fibroblast-like. But if we pre-treat them with like a purified BMP protein, that imitates that exposure to a hair-derived BMP without having to have hair. And then they become fat cells. So in vitro fat cells look like this. It's magnified. So there's all these little lipid droplets inside of the cell. It looks like just like, kind of oil on top of your soup when you drink it. So that's what they are. And this is gene expression, just basically saying, like, if you were to be an adipocyte expert, these genes will make a lot of sense to you. You'll say, oh, of course they're fat cells because only fat cells can express this gene. So that's how we know. And as I'm wrapping up here, this finding is actually fairly fundamental because it tells us that this myofibroblast state is not the final station for myofibroblasts in scars. They could go further. They could actually go in a completely different direction and they can become new fat cells, which would be quite desirable. So here we worked with a specific type of a human scar called keloid. It's this like very exuberant scar that protrudes. Only certain individuals with genetic predisposition can form keloid, but they often get cut out. So we could get a lot of tissue samples, and then we can culture this keloid scar cells in vitro. And if we try to make fat cells out of them, it doesn't work. So fat cells here labeled with this bright orange marker. So yeah, in normal state, like very few of them become fat cells. But if we would do an experiment when we culture them in the presence of human hair follicles, and we get these hair follicles from hair transplant clinics. Believe it or not, many hair transplant patients will happily donate a few hairs for research. So if we do that across membranes, so these hairs will make molecules that will diffuse to the cells, and there include BMPs. So they will expose scar cells to BMP. That by itself is enough to alter their fate, and now you see a lot of green. So they all can turn into fat cells in our experiment. So this probably means a lot for anti-scarring strategies, that suggest that hair-derived molecules like BMPs could be very potent anti-scarring molecules. It probably also means something for like aging, right? So typical signs of facial aging beyond hair growth and hair loss is also formation of this deep wrinkles. So if you're a plastic surgeon, you would know. So what causes deep wrinkles underneath the skin is actually permanent disappearance of deep facial fat that runs out of its fat stem cells. So no matter what food intake, a lot of times just older individuals would not be able to expand the facial fat, and skin around it basically sort of just collapses on itself, and that's how deep wrinkles form. So idea is that, if we could, there are fibroblasts, there are plenty of them. If we can convert these fibroblasts to new fat cells, perhaps you can reverse this to this, and many people will pay a lot of money for something like that, actually. So who would pay money for this? Examples include, pet projects of Google. It's called Calico Labs. They're infusing huge amount of money into it. There's a new anti-aging company that came about in California that's backed by Amazon money. So the question is like, what's the interest, right? So I think idea is that as humans are able to prevent death from things such as cancer or cardiovascular disorders, so that could add another decade, maybe decade and a half to our life. But you would want to have those decades to be of healthy living. So these companies work on trying to undo aging changes in various tissues, and a lot of it is linked to basically kind of reversing the age look of tissues to more youthful. So, there's a lot of articles written on it. So Bezos put a lot of his money. So, he obviously worked out a lot and, changed his image quite a bit. And now people are thinking, well, now that he has this pile of money, like he kind of wants to enjoy it for a little bit longer. So the question is, can he cheat death? And probably Altos will have an answer for that. So thank you. That's all I wanted to share with you today. The next part of the talk would be by Angie, who is working in my lab as postdoc. I think you've heard a little bit about hair, so probably by now you understand that like hair is kind of important. So Angie will tell you just a little bit more about the work that she's doing on hair. So come in, Angie. So that's your first slide.
Thanks for the introduction, Dr. Plikus. And I would like to first thank Dr. Anderson and Cummings for this great opportunity for me to become here to share my story about my journey to regenerative medicine and also some of my research work in Dr. Plikus' lab. So when I was a dermatology trainee back in China, I often encountered patients like suffering from hair loss. So although hair loss will not cause threat to your physical health, however, it can actually dramatically affect a person's individual mental health by causing great depression as well as social anxiety. And I was also deeply struck by the strong desire from those hair loss patients for better solution for hair loss treatment. So ignited by, so this clinical experience ignited my interest in finding a new solution for hair loss treatment and also led me to the journey to hair follicle biology. So back in 2017, I had the privilege to join Dr. Plikus' lab as a visiting graduate student to study hair biology. So what is a hair follicle? So hair follicle is a mini organ in the skin with a nice cylinder shape. So throughout lifetime, hair follicle can enter cyclic stages of active growth and degeneration and also rest. Also, people call these stages as anagen, catagen, and telogen. So there's an interesting phenomenon coupled with hair growth is that it can also cause melanogenesis, which can cause skin pigmentation. So if you continuously shave the mouse skin, you can use the skin pigmentation pattern as a readout for hair cycle stages, as well as an indication for hair follicle stem cell activation event. Hair growth is fueled by hair follicle stem cell, which is located in the bulge region here. When we talk about stem cell, we also talk about stem cell leash. So what is a stem cell leash? Stem cell leash is referred to as the microenvironment generated by the laboring cells, which is thought to play a very critical role in regulating stem cell behavior. In the hair follicle stem cell leash, there's a very critical component which is formed by this special cluster of cells located at the base of the hair follicle, which is called dermal papilla cells, also known as DP cells. So these special cells, they can actually dynamically send signals to hair follicle stem cell and regulate the cyclic hair growth. So you can kind of link hair follicle to a 3D printer, where hair follicle stem cell is kind of like the necessary materials you need for the printing, and DP cells it's like a CPU, which is basically regulate all the function and the process, which in the hair follicle is the hair growth. So what we found out in the lab is that the gene called leptin receptor, in short LEPR, is preferentially expressed in the DP cells. As you can see here, the red color indicates the mRNA expression of LEPR gene. They are mainly in the DP cells. So this is suggested that LEPR can be a marker gene for DP cells. So then we wonder if we can use a genetic tool based on the LEPR gene to label DP cells. And luckily, there was already a well-established Cree line called LEPR-B Cree. People often use this to study metabolism because leptin signaling is well known be associated with body metabolism. But however, this the labeling pattern of this Cree line in the skin is largely unknown. So we are using this Cree line and created a reported mouse line called, where you can actually simply check the LEPR-B Cree labeling by just looking at the cell color after a spatial staining called LAC-Z staining. So basically, if you see a cell with pink color, that means this cell has no LEPR-B Cree labeling. If you see the cell with a blue color, that means the LEPR-B Cree labeling is turned on in those cells. So as you can see in the mouse skin here, the majority of the cells are pink color, and the blue cells you mainly see in the DP cells. So this result suggested that LEPR-B Cree mainly labels the DP cells. So we then ask if we can use this Cree line to modulate the gene expression in the DP cells and further regulate hair follicle stem cell behavior. So we use this Cree line and overexpress the hedgehog signaling in the DP cells. As you can see here, based on the skin pigmentation pattern, the control mice, after P46, they enter a long period of resting phase called telogen. However, in the mutant mice, you can see that soon after P46, they enter another bout of active hair growth. So this suggested that activation of hedgehog signaling in the LEPR-B Cree labeled DP cells can cause hair growth hyperactivation. So struck by this very strong phenotype, we then asked, what is the mechanism behind this phenotype? So to be able to answer this question, we leveraged the advancing technology called single-cell RNA sequencing. And with the great help from some computational analysis tools, we were able to identify a molecule called SCUBA3 and seemed to be associated with the hair phenotype in the mutant mice. So then we wonder, is SCUBA3 really the key player in activating hair growth? So to test this, we injected the SCUBA3 protein into the mouse skin and to see if it can induce hair growth. As you can see here, compared to the control site where we inject the skin with control protein BSA, the injection sites with SCUBA3 protein showed dramatic hair growth. And also the mice with SCUBA3 gene deletion also showed delayed allergen initiation. All these results suggested that SCUBA3 indeed can induce hair growth. Based on those findings, we actually filed a patent because of its potential on hair loss treatment. So then what we're going for next? With the support from the CERN Institute, we're now trying to figure out how SCUBA3 signaling can activate hair follicle stem cell, hoping to find out more molecular mechanisms and also gain more insights for hair loss treatment. Thanks.
Max, And Inti, can you say how you got interested? Like, how did you guys start down this path to begin with? I'm putting on my Peter Donovan hat for the moment, right? Because yeah, why skin? Why wound healing? Why? I mean, you're a dermatology resident, so you know. But I don't know that for most people, maybe in the audience, but probably certainly in California, as the lay public who's interested in stem cell research, I don't know that it would be obvious that skin's a really good model for that, and why that would be interesting in terms of dissecting how stem cells work. Or, you know, was your grandmother bald and you just felt like this was the way to go?
Okay, I guess I will just go first. So for me, when I went to medical school, at the beginning I did not know where I wanted to go for like my specialty. So when I went to the clinical rotation, when I follow my mentor in the clinic, I just got amazed by how the differential diagnosis in their for like dermatology conditions, and also like how accessible or like how feasible the skin model for like testing all those conditions, and especially for like hair follicle study. Mm-hmm.
I don't think I have a choice because, you know, my name is Latvian. Plikus means that without hair. I know zero Latvian, zero, but I Googled
That's
it.
not actually
And now
true.
I know, Google it. So from now and then, there'll be like a news article in like Latvian and all Lithuanian. I was like, what is all this about? It's usually somebody without hair running around in the middle of the night and wherever, you know.
So you were destined
So
from birth.
Yes, yeah.
Okay.
If you believe in that, right? But you know, I like kind of unusual things, and skin is very visual. So there's a lot of unusual things that you know. If you go to the zoo and you start paying attention at the fur patterns, right? I mean, like it's very captivating. And it's all about hair, right? Like, so think of all of the large cats, you know, like this classic patterns that are on Gucci bags and all that stuff. It's inspired by nature, right? So like nature designed these beautiful patterns, and it all has hair at its basis. So there's something deeply fascinating about how, you know, skin has evolved. It's a very dynamic organ. It's very visual. So I think, you know, to me, that was kind of natural to be attracted to that. And I think along the way, you know, then you start doing specific projects, then you start diving deep into cell biology, molecular biology, then you realize there's a lot more to it. That's what's on the surface, right? So each hair is able to grow only because it has this really big compartment of stem cells. Then all of a sudden, because it's on the skin surface, many have quickly realized it's one of the best models for studying stem cell biology. It's just right there. So, and it turned out to be extremely fruitful for many people's careers. They built, you know, very successful careers just studying hair. They discovered a lot of things that in other systems would have not been so easy because they tucked away deep inside more. So,
Yeah. And so that was perfect, right? Because I think it's important to bring out that idea that having the right model system can really help you jump science forward in a different direction, right? You may not think about as a audience member, as a layperson, because understanding those aspects of stem cell biology now inform you about wound healing, about baldness, about potential treatments, but they also inform other disciplines in stem cell biology, right? And you have this cool model where you can watch what happens in a way that we can't do if you're studying whatever, liver stem cells or cardiomyocytes, right? Questions? I'm going to go after my people if you don't just ask. Somebody's got to have something.
Out of curiosity, do you know why? Do you know the evolutionary theory why human lost hairs?
yeah, sure. I'm quite involved in this. It's a very philosophical question in many ways too. Like, what, right? So, some time ago when we had common ancestor with chimpanzee, right? So like, you know, we split off. The thinking is that common ancestor probably looked more than like chimpanzee than us, right? So we've evolved away from that. So if you think of like, so, but the answer is more complicated because us and chimpanzee have actually the same hair density. Average human has about 5 million hairs on the body, give or take. 500,000 is on the head. It means the rest of our body has 4.5 million hairs. Where are they? Why can't we see them, right? We think of ourselves as kind of hairless. So take a magnifying glass and start looking, all of a sudden you'll see all of them, but they're just really small. So they're all there, but what they normally produce is really short hair. Unless you go to the beach, somewhere southern France, and then you start seeing this man with all the fur on the chest, then you realize, wait a second, we're not so hairless after all, right? So, but yeah, so typical humans have reduced the length of the hair, and I think it's believed to do with the fact that our early human species have entered into open plains, so savanna, and they start running a lot. So for thermoregulation, hairs became shorter, and actually compared to chimpanzees, we developed a lot more sweat glands. So we sweat way better than chimpanzees could. They live in like leafy forest in a jungle. That's what they do. But what's even more fascinating question, how come very locally on our head, hair got longer? So there was a recent paper from one of the leaders in the field who basically studied that, and it suggests to protect our head from like direct sun. And most likely the ancestral shape of the hair was curly. So something that you typically see in populations in Africa, really curly hair and very long. Actually, it's almost like a natural helmet against sunlight. So your skin, the scalp would be actually very much protected. And all other diversity of hair that you see in human populations outside of Africa, it's secondary. And color and texture, like the straight hair, all this stuff, it's all secondary to the ancestral feature that was probably curly. But it's very interesting, yes.
Joran. Nice to see you.
So actually, the curly study was just on Science Friday. They had a show about how they used a mannequin and measured the curly kind of heat dissipation, and that was. But then they were talking about the fact that once people moved north and so on, then that curliness was not necessary, and we could figure out how to not be sunstroke.
Yeah, we're writing a review together with that author right now, actually.
Okay,
so
there we go. But my question is, does this now mean in the regarding your research about the face and the fat cells, does that mean in the future we might have hair masks that we will be applying to us?
Right, I guess, okay, so you're referring to the fact that what I showed you, that hairy wounds and only hairy wounds will grow fat, right? So should you first have hair on the face to regenerate fat, right? So many people would say like, well, I don't want hair on my face. I want just this like baby fat back, right?
Like the kind you would buy at Target. You just apply and then you take off.
Oh, you mean like the Korean beauty masks? Okay, these are Korean beauty masks, okay? Like Target, they resell that. They did not invent them. No, well, the idea is that like many fat-inducing molecules would possibly be proteins, right? So this is like, it would be advanced biological therapeutic. So you would need to also apply them past the skin barrier. So our skin is actually quite impenetrable to large size molecules. So it needs to be micro-injected. So in this field, there's actually a lot of advances in microneedling based deliveries. So large size molecules could be now delivered into the skin very effectively through fairly advanced like 3D printed microneedling devices. So that's what it would take. I don't think topical mask would do it. But yeah, some form of microneedling would do it. And a lot of people are innovating in this field, and many biotechs are pursuing sort of the skin rejuvenation, hair growth induction through like delivery of some really potent molecules, often proteins, peptides, through the microneedling technology.
So I'm going to have two questions, but I will ask them in a row. So the first one is, you showed that you need a very large wound, and then only the center gains hair. So is there some distance measuring feature of cells, or is there a tension factor? Why is it that only the center gets hair and there's a periphery of scar?
Short answer, we still don't know. But the long answer is that we know some bits of this information in the field, including some of the work that Angie didn't show that she's doing. It's very complex, and it has to do with the very dynamic state of cells. specifically wound fibroblast, there's just like almost like a very dynamic map of cell states in the scar. So the same fibroblast in the center, at the top, in the middle, at the bottom versus edge, top, middle, bottom, they're all different. So they all geographically have like these domains where each cell is actually distinct transcriptional state and probably distinct epigenetic state. And answers some were there, but we still did not disentangle this whole thing. It's quite complex.
So then my second question is, Scuba 3 sounds like you've patented it. So what is are you doing CERM work on that patent? Are you doing is there a spin-off company? What's the plan, and how does that work for the audience if a UCI faculty and fellow invents something?
Well, the local
what happens?
Yeah, the local biotech picked it up, so it licensed this patent. So they are pushing for clinical trial on this. So
you don't have any in lab you can test on me?
Man, this will violate a lot of protocols. Ryan, you should know better than that. Talk to me later. You can buy it from some vendors and do it on yourself if you wish. Then you only hurting yourself at that point. Yeah.
So actually, let's go down that path for a second. This doesn't happen after hours in the Stem Cell Center. Because actually something you said about microinjection, right, in the context of restoring, like away from deep wrinkles, restoring the natural fat as a sort of a youthful treatment, reminded me of an article. It's more than 10 years old for sure. That you can Google it, was in the New Scientist, called Bones Get in Her Eyes. And it's one of the examples that I always use for why you should not go to your corner stem cell therapy thing in the mall, where basically the idea, it sounds like you're familiar with it, so maybe you can explain it better than I can. There was an unfortunate attempt to do a similar kind of treatment, right? That had a side effect of inducing bone formation as a part of a cosmetic procedure to treat wrinkles, right? And so driving cells inadvertently down the wrong path while you're trying to do this could be a potential problem. And so I don't think you want to do this after hours in the lab, but I think that would be a great story to talk about a little bit in this context, because it really does touch on an industry that's problematic in terms of regulation and management and moving forward in a safe way in this space, right?
Yeah, I think there's a couple problematic things that fly under the FDA radar, because typically this involves taking your own cells and shuffling them around in the body, so putting to the unnatural locations, or taking your own soup of molecules such as plasma, platelet-rich plasma, and injecting it in like joints, you name it, eyeballs, right? People have done crazy stuff. So FDA doesn't look at it as a drug, because it's your own body fluid, it's your own body cells, so they just essentially get shuffled around. And a lot of time, like, they're sold, by people who should not be honestly doing that as a miracle treatment for everything, right? So desperate people will fall for this. FDA doesn't currently regulate that, and that creates a lot of problems, right? So because it's your own body components, but they are just moved around. So a lot of times people get this liposuction procedure, so they'll remove abdominal fat. Then instead of being a surgical discard and just like toss it into a biohazard bin, it's often gets centrifuged, and the bottom pellet that contains a lot of fat stem cells will get injected elsewhere. Sometimes it's injected into deep facial wrinkles, and then new fat cells will form from it. That sounds good, but a lot of times it becomes very lumpy. And a lot of times there are side effects, because you're injecting high concentration of very potent adipogenic stem cells into very fine location, and that's why it's very not so simple. But it's not a drug at the moment.
I think that's what happens in this Bones Get in Her Eyes case. But the challenge was the individual who was doing the injections combined it with some other standard that was used as a cosmetic procedure that had a factor in it that triggered osteoclast formation, right? And so this woman presented like several months later with eye pain because they had inadvertently stimulated the formation of bone from those adipocytes. Other questions? Oh, perfect.
Thank you. Excellent presentations. My question has to do whether you looked at hormonal levels as variables that would affect both growth of fat and hair.
Well, of course, I mean, that's a great question. You know, hormonal sensitivity is the culprit of main form of hair loss in people, right? So androgenetic alopecia, which affects not only men, but it does predominantly, but also a lot of postmenopausal women, has to do with inherent sensitivity of cells that Angie really talked about, called dermal papilla fibroblasts. So these cells are not only relevant to mice, they're super relevant. They're at the core of the hair loss. They're the ones that express what's called androgen receptor. These are the cells that would respond to testosterone and its derivatives in our circulation, right? So currently there is an FDA-approved therapy originally owned by Merck, but now there are generics because the patent has expired. And essentially it's a small molecule that interferes with conversion of testosterone to its much more potent form called dihydrotestosterone, DHT. And taken as a pill daily, it could stall hair loss. So if a 30-year-old something that started to lose hair get onto this pill, it would not necessarily reverse everything, but it would stall further hair loss. So it's quite effective, but compliance is very complicated for many people. Daily pill, it's a lot of commitment. But it really shows you that it's hormonally driven, right? So, which presents a very interesting problem because like in normal human, you cannot get rid of testosterone. You need it for a lot of other normal function. So how could you reverse hair loss when there's all this background of testosterone, right? So you need to discover some really potent hair growth activators that could act downstream of it. And despite testosterone, they could still induce hair to grow. So there's a lot of innovation in this field because since that Merck, you know, FDA-approved therapy, which is marketed as Propecia, its generic is finasteride, there has been nothing.
And that's a long
There were
time ago for
FDA
it to get a patent.
therapies for brain stuff, for any other organ you name. There were new FDA therapies. There were 2.5 decades that passed by. There's 0 new FDA-approved therapy for hair loss. Doesn't that kind of tell you that hair is rather complicated? It's not that people didn't look at it. There's so much money to be made out of this. There are billions of dollars just waiting for somebody to tap into because this is cash-only business. It's amazing. You don't have to deal with any reimbursement, right? Nobody was able to crack it. But there's a lot of biotech companies right now that are after this, and they're using all this new knowledge that we acquired in recent years just about the deep hair follicle molecular biology, that now they're using like more educated guesses. So there's a ton of biotech, a lot of them in California, going after this problem, and I think they just want to make it big. And I think they will.
That was so fantastic. But I'm going to suggest
I have one question.
second, so penultimate question.
Okay. Hi, it was a really nice talk. I was just curious, is your lab currently interested in, or in the future, interested in studying like different burn models and stem cell-based therapies for different models of like burns?
So yeah, so burn wounds, of course, is like, you know, as you can tell, it's probably one of these life-threatening types of wounds, right? So like if you're in a car crash that like car got on fire, right? This is like often life-threatening, and we're talking about deep and large surface wounds. These are extremely complicated to treat because you're also up against very short period of time, right? So recovery from large surface burn wounds is extremely complicated. We're not pursuing burn wound model, but the healing of burn wounds is actually quite different from what we study. We study like excisional burn wound, which would be probably the closest analogy to this in real world, is like, you know, large size trauma where like chunks of skin get physically ripped off and you have just a gaping hole that needs to be filled up. Burn wound is that natural piece of skin is still there, but it has been cooked by fire, right? It's coagulated. So the way you heal that versus the gaping hole is actually quite different. Early processes differ. We're not going after that, but I do admit that this is a very big biomedical challenge. So there are companies that produce artificial skin. It's not really skin. It's just like, this like ECM, extracellular matrix constructs with some cells on it that they would place on these wounds. Other than that, it's treated by skin grafts, which is never pretty, actually.
Last question. Zingyi, I wanted to know what are you going to do next? What is your career goal? And how do you get there?
Okay. So I think I wrote in the personal statement when I applied for the CERN clinical fellow. I mean, that's always my lifelong career goal. I wanted to become a physician scientist. So at the beginning so in China, the medical education is a little bit different from the US. You actually enter medical school right after high school. So at the time, I thought I just want to become a doctor. I wanted to cure patients. I wanted to make a diagnosis and make their life better. So that's what I thought. So when I entered medical school, I started to like shadow in those physician scientists where they actually do both clinic and also research in the lab. So I just feel, oh, this is cool. You can actually make more significance just other than just being a doctor. So you can use your research to answer the clinical question, and then in return, you use your research to benefit the patients, other than your own patients, also the patients worldwide. So yeah, that's my career goal. I wanted to become a physician scientist. Yeah.
So that was great. I'm going to put you on the hook for one more. So whatever. Director's prerogative. Just because that was such a nice segue into talking about training and training opportunities, right? So I wonder if I could ask you to say just another word about the CERM Scholar Training Program and how you think that has helped you. It's one of the 4 training grants that we manage here at the center. So.
Yeah, definitely. I think the CERN training program has helped me a lot. I mean, since 2017, I joined Dr. Plikus' lab. I learned a lot of knowledge from him in terms of cell biology, but I actually did not have that much clinical exposure. So, so after I got a clinical fellowship from the CERN Institute, I actually got a lot of opportunities to like talk to those physician scientists, including like faculties at UCI, at Durham, department at UCI. As you know, I'm interested in a career in dermatology, dermatopathology. So basically like more focused on the diagnosis and also like figuring out the pathogenesis of skin disease. So the CERN fellowship really got me into, got me into opportunities to talking to those faculties and really get a lot of perspective from them. And also there's a very critical component of clinical fellowship is that you actually have to establish a clinical mentor. So with that, I actually got an opportunity to talk to the dermpath expert at UCI, Dr. Bonnie Lee, and she actually served as my clinical mentor. I really hope through this program I can learn more in the future and really like achieve my goal to become a physician scientist eventually. Mm-hmm.
So I just want to thank both of you. That was just spot on tonight. Thank you so much for your time and for participating. Congratulations on all of your work, and please join me in thanking our speakers and saying good night.